Today I discuss about a special topic on chromosome, which is known as chromosome banding. All of you know that banding or band is an important part, or it is required to specify a particular region, and this banding can be used in different cases. Whereas in chromosome, this is not also an exception, and in chromosome we use to form a band, or we use to develop a band to recognize the specific region or specific sequences in the chromosome. And well, in this band formation, the band appears as a dark or light with various banding property, and there are so many bandings are present. That's why different types of bandings can be developed or adopted. In 1958, Kasparson et al. Published the first paper describing the use of quinacrine mustard to stain the chromosome, and this is also a chromosome banding, and it reveals a new era of chromosome banding. The Paris report 1971 was the first attempt to provide the nomenclature on the basis of chromosome banding. The nomenclature of the chromosome banding mainly based on the different chemical utilized to develop to form chromosome band. So this is the why we study the banding pattern, because I have already told that band reveals or band signs a special region or specific region, specific sequence. And in chromosome, if we develop different banding patterns, so we can identify the structural organization abnormalities in the chromosome, and these abnormalities, the structure sequences which cannot be visible in the routine analysis, so that by the banding pattern we can identify the abnormalities or specific sequences in a chromosome. Next, the classification of banding. I have already told that the classification banding is based on the utilization of chemical agent, whereas it is also based on the nucleotide sequences which are present in the chromosome. And we know that there are four different nucleotide sequences. One is guanine, cytosine, adenine, and thymine. And in case of DNA, the guanine form bond with the cytosine, and the adenine form bond with the thymine. So some regions with a GC rich and some regions with AT rich. So based on the GC and AT rich, we can classify the banding. And another one is the, we know that in chromosome there are two different regions. One is the euchromatin, and another one is the heterochromatin. Of which heterochromatin, there are two types of heterochromatin: facultative heterochromatin, that means which can be converted to the euchromatin, and another one is the constitutive heterochromatic region. So a band can be formed in the constitutive heterochromatic, because constitutive heterochromatic region is a condensed region, and this banding technique can be developed in only metaphase chromosome, because we know that the metaphase chromosome is the most strong and rod-shaped condensed structure of the chromosome. So to fix a banding, the metaphase structure, metaphase form is very much important. Next, come to the banding technique. There are four different banding technique. One is the quinacrine. Next is the or Q banding. Next is the G banding or Chimsa banding. NOR banding, nuclear organizer N banding, and C banding, centromeric banding. Depending on the nomenclature, we can say that the quinacrine and Chimsa, these are the chemical compounds, and NOR, that means the nuclear secondary construction, that is secondary constriction of chromosome. Basis on the secondary constriction, we can develop a band. And another one is the centromeric band, that is chromosomal centromere region can be formed band, which is C banding. And these are the different sequences: 1958, 1971, 1973, and 1978. Next, come to the Q banding. So there are different way of banding technique: Q, G, N, and C. let's come to the Q bending technique. First, we take the chromosome. Actually, this is the metaphase chromosome. Then stained with quinacrine. That is, quinacrine is the chemical which is used in the Q bending, and it is subjected to under the UV light. Then the bending pattern is formed basis on the nucleotide sequence. One is the AT reach region and another one is the GC reach region. In case of AT reach region, there the dark stain is formed. That means quinacrine stain the AT region. Whereas the GC region is not stained by the quinacrine, that's why these regions becomes light in color. And AT region, which is dye and fluorescence stay suited in heterochromatic region. By the quinacrine, we can identify the adenine and thymine region of chromosome. Next, what are the advantage? Actually, advantage of Q bending is this is the very simple and versatile, and it is used in all the organisms, and Q bending is used where the G band is not accepted, because G band is not accepted in all cases. And we can study the heteromorphism of chromosome. This one is very important. We can study the heteromorphism of chromosome in Q bending. Heteromorphism means the functional, non-functional part of the chromosome, and by this bending we can identify the functional and non-functional part. Next, what are the disadvantage? Disadvantage here, this is the photo degradation. It is a very disadvantageous character. Chromophore absorb light to a particular wavelength due to the chemical bond form, and sometimes also UV light breaks the chemical bond in between the A and T. So sometimes the AT region is not stayed with the Q band. So these are the very disadvantageous character. Next, come to the Z banding. G nomenclature based on the GIMSA, where the chromosome is treated with trypsin or urea or protease. Why we have to use these to denature the protein? And after the treated with GIMSA, that's why it is known as GIMSA banding or G banding. Then the these GIMSA interact with the DNA and with thiazine and eosine components of stain brightness sulfur rich region. After that, a band is formed, and the methyl azure, methyl violet, methyl blue, or eosine. These are the different brightening stain. Next, what are the advantage? Used in identification of band rich in sulfur content. This is also used in gene mapping, and also this banding is very important in the identification of chromosomal abnormalities and disadvantage. Very interesting. This banding is not used in plant. So why the G banding is not used in plant? This is a very significant question, because if we see that the chromosome of human is 2.3 times shorter, but plant mitotic metaphasic chromosome is 10 times more shorter than the human chromosome. So can you imagine that plant chromosome is how much shorter? Hence, it is difficult to demonstrate the arrangement of bands at the level of saturation with GIMSA. That's why G bending is very difficult. Next, N bending, that is the NOR bending. That means this bending form a band in the NOR region, nucleolar organizer region, constriction region of the chromosome. First of all, the chromosome is air dried and then treated with a 5% trichloroacetic acid and kept under 95 degrees centigrade for 30 minutes. Then treated with zero point 0.1 normal ACL and treated with 60 degrees centigrade for 30 minutes. Then banding pattern is formed the non-histone protein of NOR region. What are the advantage? Used in identification of secondary constriction of chromosome, used in identification of the nucleolar organizer region, superior banding pattern for plant, very very important, superior banding pattern. Next, C banding. centromeric banding, where the chromosome is treated with alkali solution. This alkali solution responsible for the DNA denaturation. After that, washing with the sodium citrate under 60 centigrade and kept for 30 minutes, then stain with the Giemsa solution. So here the Giemsa is used, but the pattern is different. And these washing with sodium citrate solution responsible for the repetitive DNA renature, but unique DNA do not renature. Okay. Next, after the treated with Giemsa, the banding pattern and heterochromatic region is developed. That's why the we know that the NOR is the heterochromatic region. So C banding is important in the identification of heterochromatic region as well as NOR nuclear organic region. Next, what are the advantage of C banding? Identification of chromosome, particularly in insect and plant. In plant and insect, identification of bivalent and diakinesis using both centromeric position. Paternity testing. So this one is very important. Paternity testing. Gene mapping. So we can form a gene mapping, make a gene mapping by using the C banding technique. So how can we represent the chromosome band? We know that in chromosome there are two arms. One is the short arm and another is the long arm, depending on the centromeric position. And short arm is designated as P and long arm is designated as Q. So we can divide the regions by using the P and Q symbol. We can see in the P1, P2, Q1, Q2, and the high resolution subband can be also subdivided or seen in the blank band. And if we look at the picture, so this is the short arm and this is the long arm, and this short arm is designated as P and the long arm is designated as Q. And if we see that this is a band, this is there are different types of band which is formed in the chromosome, and they are classified on the basis of the short arm and long arm. So ISCN International System for Human Cytogenetic Nomenclature. Each area of chromosome given a specific number, lowest number closest to the centromere. So this one is closest to the centromere. Highest number at the tips, that is the distal centromere. So these are the different nomenclature or symbolic representation of chromosome banding. P for peptide arm, that is short arm, Q for long arm or QE arm. And here if we see that the band P11, this is the 1 1, not 11. So P23, not 23, P23. So this is the nomenclature or identifying way of chromosome banding. This we can see here the comparison between Q, G, R and C. So in Q, quinacrine is used and chromosome arm mostly 80 rich DNA is stained. Effect under UV light, distinct fluorescence banded pattern of each chromosome. G banding, Giemsa stain, mostly repeated 80 rich DNA as well as G series DNA and distinct banded pattern for each chromosome. Same as Q banding pattern except single additional band near centromere of chromosome. R banding, variety of technique, chromosome arms, mostly unique G series DNA. Reverse banding pattern of the observed with Q and G pattern. This is the reverse of Q and G. And C banding, variety of technique, uh, centromeric region of each chromosome and distal portion of Y chromosome, because we have already discussed that C each banding is stained by the Giemsa. So if this is the largest band usually on chromosome 1, 9, 16 and Y chromosome. 7, 10 and 15 have medium sized bands. Size of C bands highly variable from person to person. So this is the comparative analysis of chromosome banding. So in this lecture I discuss about the introduction, different pattern, types, process and their advantage, disadvantage as well as the comparison in between the different banding pattern. So if you have any question regarding this, you can post in the comment box. Thank you.